Okay, so I am going to talk very fast, and I've learned as I get kind of towards middle age, as many of us who've been working together for our whole careers, I've gotten a little bit philosophical. I'm going to take a break. I was figuring it was kind of the afternoon part. And in addition to just thinking, I'm sure there are people I have left off this list. But I just want to say that I think, as Angela mentioned, we've gotten a bit really more structured in the clinical trial working group. And part of it is because I think we've gotten to the point in the last year where really CBTN was ready to start making these connections between the clinical data, the biology data that's been generated, and really just, I think we've seen that with an explosion of some projects, of which I'll mention a few examples. But I was going to take a step back and just a word about inspiration. I think almost every talk I've given, I realize I have talked about something I've been watching with my kids. And this is an example that I recently, because for whatever reason, we now have Disney Plus as well as all the other streaming things, heard this great story. And if you go 100 years back, the leading cause of death in children was not brain tumors at that time, but what we now call vaccine preventable disease. And in thinking a lot about what we can change and what we can't change, I focus my research on treatment as one of the modifiable factors that I think is the bridge between understanding biology and survival. And some of that bridge is, the inspiration and the scientific discovery, and some of that bridge is just the work. And I'm very inspired. I actually started my life as a cat person and then married someone who was allergic. And as many have seen when I don't have a virtual background, I'm now a dog person. This story is great for many reasons. One is that this is Togo, who is called the hero of the gnome serum race because he was the lead sled dog that was responsible for bringing a serum to cure children in Nome with diphtheria over a really tremendously treacherous course in Alaska. And this is what inspired now the Iditarod. I know about this story because of a pretty great Disney movie, which, if you can believe it, it's hard to believe that the actors aren't related, but the dog Togo is actually played in this story by his great grandson, 14 generations removed. And I think some of the things that are quite inspirational about this is that the story is the parts of the story that are the most unbelievable are actually the things that are true. And I think a lot of the things that we do and we put our efforts towards are not the things that get recognized. And so the other part of the story that I love is that Togo ran 200 miles when the average other sled dogs in this relay ran about 20 to 30 miles. And then at the end, they put up a memorial in New York to Balto, which was the dog that did the last 30 miles. And so there's a couple things I think you can learn from this. One is that, the race we're currently in to cure children is really both a race and a relay. It's very important to go as fast as we can, but it's also important to hand off where we have different expertise. And I think one of the things that's been really inspirational about CBTN is that we can each focus on the areas where we have our expertise and then hand off to people who really speak different languages. Ultimately, the goal here is to cure children and not to get credit for our work as single people, but that we all do this together. So it's a little bit roundabout, but I think I look forward to the time where we can tell the great stories about how people used to die of pediatric brain tumors. One of the things, getting back to sort of where we are with the data, that I'd like to mention is that the clinical data projects, when I spoke at the last meeting, I requested that everyone from the CBTN and PNOC working groups request data. And I'm really happy to report that we've actually seen a ton of data requests since the last meeting. There are data requests currently being worked on in areas of low-grade glioma, ATRT. There's some upcoming discussion of using the ETMR data, craniopharyngioma, diversity. There's a really neat project that was based on looking at a specific mutation in IDH1 mutant tumors and then reflexing into getting the clinical data that was relevant. And I'm going to talk just a little bit about medulloblastoma because it's something that, as Angela mentioned, I do focus on with both of my hats, both in terms of clinical trials work, as well as on the CBTN project. So I'd like to use this example of medulloblastoma as an explanation for what I've, I think, said at many of our meetings, which is that the goal of this is that we could ask and answer questions in clinical data in 10 minutes by crunching data that already exists that have taken us 10 years of clinical trials. And so this is just an example of clinical trials focusing on the youngest children with medulloblastoma. We're really, over the last 20 years, international efforts in clinical trials have been to find ways to treat them by avoid radiation. And what we have done as a community is really increase the intensity of different cytotoxic therapies that we've given, because in the long run, those still are less detrimental to childhood development than radiation. And so how do we tell if we're doing project progress here? And I think this is an example of a 10-year trial, which had, I think, really a remarkable result. The trial was designed before the subgroups of medulloblastoma were understood. We now know that young children with medulloblastoma generally have one of two molecular subgroups, either the hedgehog group, which tends to do well with very intense therapy, or the group 3 medulloblastoma group, which historically was described and not that long ago and is often still quoted as a very inferior outcome subgroup. People often still quote about a 30% survival in this group, but what we saw in the recent trial is that that was actually the group that has continued to respond to increased intensity of therapy. And so the survival without relapse was 70%, and 80% of the children with group 2 medullo in that trial are alive. Now that percentage is a little bit frustrating because when you break down all these subgroups, again, a 10-year trial, we're actually talking about 10 kids. And so saying 8 out of 10 kids are alive is very different than saying 80 out of 100 kids are alive in terms of how sure are we that this is really useful. This is statistically significant. I think there's still clearly a question of, how believable is this, and how do we change our practice, and how much do we emphasize this? This is a cartoon I found about, it says things got really interesting when the statisticians started doing rounds. So patient asked, does it work? And the answer is, it depends what you mean by does it and work. So how do we take all of the kind of data from clinical trials, and how do we add additional data to try to say, how should we be treating patients, and how do we actually impact what the goal is in cure children? So should we base the next 10 years on essentially 10 children? What other factors, and people do say things that I like to disagree with about how much does it still matter knowing biology, extent of resection, metastatic disease, things we can change like the intensity of treatment. How does the regimen that we ran in a trial here in North America and Australia compare to regimens that were being run in Europe? And are we at a point where we can feel comfortable dropping radiation therapy? So this is the CBTN cohort for a similar group of patients. And fascinatingly, none of the kids in this cohort, mostly because our institutions were involved in some other trials as well, that I pulled, were actually on this trial. And this is about 70 kids, none of whom were on the trial I just mentioned, but all had the same category of disease, young children with medulloblastoma that you would ideally treat without radiation or with limited radiation. And so what kind of things can we see compared to the trial? One, metastatic disease still matters. Extent of resection actually still matters, which is still something I fight about a lot. Do you take kids back? But there are certainly more kids alive who had a complete resection, some of which went back for second surgeries to get there. And then what if we try to set up the analysis like the 334 study? So this is just looking at kids. There were about 45 of them that were treated similar to the 334, either with or without methotrexate. And actually what's fascinating to me, and I think this means our data is better than a lot of retrospective cohorts, because it's not truly retrospective. We're putting it in the same way we put in clinical trials. You can see the same results. There are actually more kids in the CBTN database who were not on study, and we see this improvement in survival with methotrexate. And in fact, when you look at the one on the right, I took out all the kids who got even any radiation, because we were using strategies where we gave focal radiation or lower dose radiation. And if you look at the kids that were just treated without radiation at all, we can see the same thing, a clear benefit to just treatment with methotrexate or without. And again, more kids that we have in the CBTN database than were ever on study. And I think this is not to say to do this instead, because the reason this line is short and it only starts at 3 years ago, because we weren't treating kids this way till we got the early results of those studies. But a year from now, this line will very likely be out to here, and 2 years from now, it will be out to here, because we are following these kids in essentially real time by the CBTN institutions. And if it weren't for efforts like CBTN, all of this data and these kids that are twice as many as were ever on trial would be lost and not be able to be used by people like us who are trying to make clinical decisions. And so I think this is really powerful. That's the experiment I've been wanting to run since CBTN started, actually. So I was really excited to be able to do that last night. I want to reemphasize there is data available. If you are thinking of trying to ask a question, please do use the help desk, contact me or Angela or Cassie from the working group and say, how could I ask these questions? What would be available? I think there are some really important upcoming next steps, some of whom I think people are going to talk about and Adam alluded to, which is that it is now really essential and more common that kids are getting clinical sequencing data, and how we integrate that. Is that clinical genomics, it's both, but how do we put it in and make sure we're putting that kind of data in so it's also usable in real time? And then also our efforts to connect the CBTN data to other data sets with clinical trials and in Europe and all over the world is really important, because that is how we're going to compare and help all the children in the world. So apologies for some of my non sequitur parts, but again, I just find it very inspiring to be part of this group and part of this meeting and try to do my part as one of the sled dogs. So with that, if there are any questions, I'm happy to take them now or later.
Yeah, thank you very much, Sarah. That was wonderful. There's a there's a question in the chat. And it says, are treatment data on the CBTN patients publicly available?
Yeah, that's a great question. We are working with how to do that. And so I think they're not, I will say actually, as David was talking earlier, the survival information is available. And I actually pulled, while David was, and I'm not very good at this, but while David was talking, I pulled that same survival curve off of the web, and it really is the same. So the survival data is up there. We're working on how best to get the treatment data up there. And it's a little bit free-handed, although what we've done is basically, as oncologists, we follow protocols. And so rather than sorting through long lists of descriptions of data, we have they were treated according to protocol S, which is generally understandable. So it's not public yet, but it's available and been given out to people who ask for it. And it's mostly, I think Allison and the team can probably talk to that. We're working on how to make it just self-serve, but right now it's not in a checkbox form so much as a lot of freehand. So that's actually a big effort of the clinical data working group, I think, where we can try to get that. But the treatment data is really what's unique about CBTN. And yeah.
No, I just want to say, Sarah, I always get inspired whenever you present. And so thanks so much for that presentation. To the question, I just want to clarify, because I think it's important, is all the data is publicly available in the sense of it is made for public consumption. What Sarah's pointing is that some of the work that Alison Heath was talking about yesterday, for example, and the way that we have to structure and make the data queryable is still ongoing, partly because, as Alison mentioned, clinical data is not A, C's, T's, and G's. It's much more complex in ways that we want to structure it and make it into that fashion. But irrespective of that, what I think Sarah's work shows is that we can ask those questions now. So any question that people have in mind, we want to begin starting that process with you to interrogate that data, and that'll help us understand even how to structure that data for queryability longer term.
Adam, this is Angela. I know we're behind, but just want to capitalize on one more comment, which is one of the things CBTN has been focused on, and you've seen it highlighted yesterday and today, is solving solutions that sound easy but don't actually exist. And so making publicly available, curated clinical data that is longitudinal and captures all of the different elements that Sarah mentioned, and additional ones is not easy, and it's something that we are very committed to making it available. We've tried a couple of different portals or platforms that were commercially available or homegrown. Hasn't quite manifested yet, but we're getting really close to being able to do it with Allison's group. And just as Sarah said, the overall goal of this will be at some point, just like what David Higgins was showing for the genomic data, that at some point that clinical data in detail will be available in that kind of same way.
And to your point, Angela, the recognition of the challenge is something that the NCI has also come to terms with. This is partly why there's such a heavy-handed effort in the Pediatric Cancer Data Commons initiative to establish standards, as well as CCDI, and they're using CBTN data as a use case in those efforts. But again, Sarah, thanks so much, and for all the clinical working group members. It's such an exciting frontier.